All right, hello everyone. It gives me great pleasure to introduce Professor Songi Han, who will be talking to us today about her work on the tau protein. Songi received her PhD degree from Aachen University. She then pursued her postdoctoral studies at the Max Planck Institute in Mainz, and then a second postdoc as a Alexander von Humboldt Theodor Linden Fellow at UC Berkeley. She joined the faculty at UCSB in 2004, and she was rapidly promoted up the ranks to full professor. Songi's work is very innovative, and you can see that by the awards that she gets that highlight the very unique spin to her research. And I'll just highlight two recent awards, the NIH Innovator Award and the Biophysical Society Innovation Award that she received just a year ago. So Songi is an expert in NMR and EPR and spectroscopy in general, and she has really done some foundational work in tau assembly, both looking at tau conformation, tau aggregation, and tau liquid-liquid phase separation. And Songi, I'll turn it over to you, and I'm excited to hear your talk.
Thank you, Joan, for your introduction, and also thank you to the organizer. This is one of my top favorite Zoom series, so very happy to be part of it. So I would like to talk about methods and approaches to tracking the dynamical conformational ensemble of tau along its aggregation pathway. So from the title, you can see the focus is on the tau protein. And even though many in the audience, of course, know what the tau protein is, a brief word about this protein. It's a microtubule binding protein. It has 6 isoforms. I'm showing you here the longest one from residue 1 to 441. There's an N-terminus, a polyproline-rich domain, microtubule binding repeats 1, 2, 3, 4, at least for the long isoform. The 4 are isoform, and the C terminus. And it's an intrinsically disordered proteins. Now, under adverse conditions, not entirely known what the exact starting point is, tau protein falls off the microtubule and stacked to fibril and deposited as neurofibrillary tangles. Now, tau aggregation is particularly important to study, and the pathway particularly important to understand, because the deposit in the brain are clinically diagnostic of many important neurodegenerative diseases known as taupathies, including Alzheimer's disease, chronic traumatic encephalopathy, to name just 2 out of the many taupathy diseases. And in fact, often only post-mortem analysis of the tau fibril will show definitively the diagnosis. Now, many in the field were really excited when the breakthrough cryo-EM structure of post-mortem tau fibril came out, the first one in 2017, because the structure was different from what has been imagined, much more intricate. And then a few years, within a few years, 4 structures were published from Alzheimer's, PID, CBD, and CTE diseases, all from post-mortem human tissue. And what these studies from Goddard and Sherris have shown is that unique tau structures represent each taupathies in folding specific ways. And so this is important among others because it shows us something that many already knew, but this is definitively showing how important the pathways are, because the specific structure that is formed needs to be replicated, since many pathways might not be disease relevant or even non-toxic. Now, in the and many reasons why the pathway is important, but one way to summarize that would be, in order to develop therapies for example, small molecule compounds or antibody, those development require the ability to replicate the disease phenotypic tau fibril from naive tau, a goal that hasn't been achieved yet. There are many unknowns. For example, it is clear that tau has prion-like properties in the cell as well as in mouse models. But what entity exactly initiates and propagates tau aggregation is not known. In what way exactly post-translational modification, fragments, or cofactors help along this process is highly debated. And ultimately, at the end of the day, neither rationally nor empirically, meaning by trial and error, has the dynamic pathway for specific structures been achieved so far. So far, all these end structures that represent a disease come from human postmortem material. So to date, no disease-specific fibrous structure has been replicated nor validated in a laboratory outside the patient's brain tissue. Still, of course, current approaches to the medicinal compound screening go on strongly, and they rely on the so-called strain hypothesis that seeding with a parent tau fibril faithfully propagates a strain onto daughter fibril. Now, the structural basis of this hypothesis, whether and how much and in what way the daughter fibril adopts what part of the structure of parent isn't known. But there are a lot of higher level evidence and support for the strain hypothesis to have a strong basis. For example, here, when taking material from the Pick's disease versus the Alzheimer's disease and inoculated with a mouse model, and I'm just highlighting here one of the recent studies by Virginia Lee's group, it is clear that some property in terms of deposit as well as pathological behavior is replicated. Now, the strain hypothesis finds furthermore support from the developments by the Mark Diamond group and the Prusner group that inoculate what they call biosensor cell with human tau material, or in some cases, some studies also mouse material, and find a cell-derived daughter seeds to be potent in strain-specific way. But when thinking about how and why does it even work at the molecular level, there are many questions. For example, the incubator tau material is typically K18 with those P3ONL and V337M mutations that are nearby the R2-R3 transition and the R3-R4 transition. How does it work is really unclear because, for example, this fragment doesn't include the entire core, for example, of the AD fibril. So this kind of question exactly, maybe perhaps part of the structure is propagating, perhaps that is already sufficient, but these are exactly the kind of questions that require structural biology studies. And now at the current state of the art, that requires in vitro studies, in vitro aggregation in order to do structure studies, except of course for cryo-EM when human brain-derived materials were used. So in vitro studies, there are several techniques that have been successful, of course, cryo-EM. Also, this has been shown used for resolving the heparin-induced fibril. In this case, the conclusion by the Goddard and Sherry's group have been that there are heterogeneous fibril present. Solid-state NMR studies by the Bill DeGrado and Mei Hong group have shown structures that converge under certain carefully prepared condition that replicated part of the CBD structure. And micro-ED studies that have resolved structure of small fragments of tau. But a technique that I would like to focus on today is double electron-electron resonance. Now, our group have used that, but also I should highlight that Martin Margitay group has done very important and very beautiful work using this double electron-electron resonance study. Now the difference is that when you the difference is that cryo-EM, solid-state NMR, and micro-ED are techniques that are very powerful but require already converged structures. In other words, you already have to be successful in having a good fiber. And then it's, of course, a very powerful technique. But especially in tau aggregation, we are learning that we need a way to do structural biology that captures the whole process from the IDP state and even when the pathways don't converge, knowing that part of the structures has formed or the structure is heterogeneous or two different structures are present are all part of the important information. And so here, double electron-electron resonance spectroscopy, which is an EPR technique, is very powerful. It relies on a pair of spin label, typically nitroxide spin label, that we place flanking specific segments, for example, let's say around certain segments in R3, or some of the hairpin that has been postulated between R2 and R3, and in some cases end-to-end if the protein is a little smaller. So then when the protein, for example, in case of tau, adopts a whole ensemble of conformers, DEER captures that probability distribution of these distances. And if it's an IDP, it will yield a broad shape. And when a distinct fold is adopted, as illustrated in this cartoon, then the segment end-to-end distance that we have chosen carefully, depending on what structure we expect, will yield then a much narrower distribution of distances. And here, the P of R, even the width or the number of peaks, of course, contain information about the homogeneity of the fibril. Now, I have to also emphasize that typically we only label about 5%, or in some cases 10% of the protein, and 90 to 95% is the wild type, to make sure that the signal comes from intra-protein distances and distance distribution. We are not, at least currently, interested that much in the inter-packing, in the intra-shape. And also for those who perhaps do single molecule FRET, I should emphasize that EPR captures signal from about 10 to the 14 spin label molecule. So the statistics for P of R is very large. So it's sufficiently large to really capture quantitatively the distribution. So let me just show you one of the DEER studies. Was actually just published a few days ago, and it illustrates a point I want to make nicely. So I want to talk about that work just for one slide that we have done. So DEER can reliably capture the ensemble structure of an entirely unstructured, unstructured excluded volume polymer, for example, on PEG. So here we end-to-end spin labeled PEG at varying molecular weight. And you see here the time domain dipolar signal that DEER measures, dipolar signal as a function of time, and very carefully done inversion routines can give us the P of R. And you see here, as you go from a small molecular weight to longer, how the P of R gets broader and broader. And when we then plot in a classic way how polymer physics community does it, the mean end-to-end distance, the scaling exponent with molecular weight fits to 0.588, except for the smallest one. There is a good reason for that. So it's showing what we know, that PEG is an excluded volume polymer. But P of R from DEER is so much more than delivering the mean end-to-end, because it's a whole shape. And that becomes particularly important for the study of tau when we expect emerging shapes to come out that we wish to capture. So let's see whether this end-to-end distance distribution or segment to end-to-end distance distribution, how we can apply that and what value it has. So I'm showing you here an example of a fragment, 295 to 313. It is flanking the region between R2 and R3, okay? And we take the wild type and the P301L mutation. This is one of the most used mutation to mimic disease or to induce aggregation-prone properties. So when you take those two tau molecules and measure the distance distribution, you see that in the monomeric state, they're nearly indistinguishable, and they're very broad. They truly are IDPs. And what kind of value can we get out of from this kind of measurement? Well, it is still really, in some sense, surprising, because the two protein, one, the wild type that we call HP1, the P301L, have very different aggregation kinetics. And the mutation not only aggregates faster, but the final fibril count is much higher. And in some sense, you can see that also from the TM, it's a much higher fibril forming potency. So it looks like in this case, the initial conformation doesn't capture that propensity. Something else is going on, and I'll talk about that towards the end a little bit again. But the reason why we called it HP1 and HP2 stands for hairpin, because as part of the CBD structures, that fragment would form a hairpin if it was adopting the CBD structure. Now, if our fragment would adopt that hairpin structure right away, I would be showing you the fibril PFR. That's not the case. All I can say at this point is that the fibril structure of PFR is distinct from the monomer. There are distinct peaks that show up, but I can also say they are not one, but there are multiple peaks that we see, and we need to figure out how to achieve convergence. Now, since right away getting the hairpin is something we are still working on, let me show you another example of segment flanking and studying the distance distribution. We can take the R2 and R3 hexapeptide region, VQIINK and VQIVYK, which is one of the two most potent beta sheet packing segments. In all of the structures published so far, both postmortem human as well as from the in vitro studies, each of them have this R3 peptides included, and some also the R2 in the published structures. So it's a good one to think about. We have labeled then those fragments end-to-end, R2 as well as R3 separately. And here I'm showing you the computational work that Joan Shea's group has done of that fragment in an IDP state, when we expect for it to stretch out to form fibril. And you see the computed PFR of the monomer. Of the dimer, it already extends towards longer distances, but it's also broad. In the fibril, you see a much more extended structure is adopted, and the width is narrower. And let's see what the experiments show us. So those are experimental PFR measurement measured by DEER. And you see that, and so here, these are tau where the R2 or the PHF6 star region has been flanked by a pair of spin label. And the second case is where the PHF6 region is flanked by a spin label. In black is before, and in red is right after aggregation is initiated. And you see after like a couple minutes of heparin addition, you see the distance shifting already. And within about 10 minutes, this PHF6 and 6 star region is entirely extended, at least as much as it is after 12 hours upon heparin addition. So this was an interesting finding that we have reported on a couple of years ago. But the important part is that, yes, we see a conformational extension, and that preparation of the tau to stack in the form of conformational extension of this region happens within about 10 minutes, which is much, much shorter than it takes for the maturation of the fibril to occur. In other words, the whole ensemble already adopts their structure, and then it takes time for the fibrils to anneal. But this PHF6 and 6 star region, especially in light of the cryo-EM structure, are not discriminatory. All of the fibril have this segment, and so those would be not very effective regions to look for when we are interested in different fiber types. So here I'm showing you an experimental design. Shown are the PID, AD, CTE, and CBD fold of tau. And you see here various sites that are labeled, for example, 315, 373, okay? So as you see 315, 373 in PID, or in AD here, 315, 373. And the point is that if these different ones and if these different shapes are adopted, then the distance distribution would be discriminatory of, for example, AD versus CBD or CTE, okay? When you look for 351, 373, let's look here. This is a particularly good one because those pair of sites have the spin label on the exterior of the fiber if those structures formed. And you see that if you had AD, CBD, CT, or PID fiber, these distances really would be good markers to tell us, yes, this fold or that fold has formed. And so this is currently what we are working on with seeding material. Now, when the cryo-EM structures came out initially of AD, we got, of course, really excited. We labeled this site, we induced aggregation with heparin, and wanted to know what kind of structures are formed. And in red and blue are shown the structures from the PDB structure, and in black is shown what we actually measured. And with the, after talking to Michel Goddard, we decided to even publish that study because we really worked hard to try to figure out what structures form. But what we can say is without particular methods of using a cofactor, using a seed or certain procedures, if you just initiate aggregation by adding tau, even after careful preparation of the fibers are quite heterogeneous. So the question then is, we need to find ways to achieve convergence, right? It's not that you can just initiate aggregation and a unique structure or even unique structures would be developed. Oftentimes is really just very heterogeneous. So the factors that have been postulated so far by us and others include the use of cofactors that include RNA or other proteins, post-translational modification, protein purity has been postulated by DeGrado and Mei-Hong's paper, fragmentation is an important concept, and seeding as well as liquid-liquid phase separation. So I'll talk about the last two because this is where we can propose some at least advances. And I will first talk about seeding, and that will transition into the use of LLPS potentially. And we decided to focus initially on seeding because that is the approach that therapy development is based on, is seeding, both with mouse model as well as cellular model. So this was a collaboration with Ken Kosick and Jenny Rauch. They have helped us generate from the tau AD model mouse brain. We have received paired helical filament material, and we then went ahead and did seeding studies. And what we have seen is something that other experts had told us will happen, namely, when you take the PHFs from the mouse brain, in the first generation, it's very potent. But when you take then the seeded fibril and would like try a second generation seeding round, somehow the potency drops by a lot, okay? So something is there in the PHF material that is not there in the second round. And so long story short, there is some sort of factor that is important. And so cofactors especially with the cryo-EM structure coming out, they show that there's stoichiometric amounts of some cofactor present, for example, in a CBD and CT fibril. But here we also knew from experiment earlier that something is involved that is a cofactor. So which cofactor might be physiological? And so we got inspiration from the Kozik group and Mei Zhang, because they have shown in fact, in a joint publication together, that RNA in particular, for example, tRNA, naturally bind to tau. So they might be natural cofactor that we can use. So we did the seeding studies, and long story short, I think what I can say is that when you seed just with the just with the seed, there is no aggregation. When you seed just with the cofactor, there is no aggregation, or the amount of cofactor is tuned on purpose to be mild. So RNA by itself at small concentration doesn't induce, at least not effectively, aggregation, and the seed at the percent we used also by itself cannot induce aggregation. But when we add a cofactor that in and of itself cannot induce aggregation, then we have seen that using the brain extracted seed that again in and of itself cannot induce aggregation, with the cofactor first generation as well as second generation, we see efficient aggregation. Okay, so we have reported on this idea that we call the three-player game, that we need the fibril seed as well as the cofactor to have potent aggregation. It was really exciting to also see, even if you take heparin fibril seed, of course, knowing that those are heterogeneous fibrils, again, at the amount that the seed itself is inactive, or with cofactor that in and of itself is inactive, all three together can induce efficient aggregation. Okay, the same thing I think is shown here in this histogram. And so then we again looked at PFR with DEER. And in green, you see the heparin fibril, as shown earlier, just a very broad distribution, even though the mean distance is extended more towards 4 nanometer, which is the fibril shape. And was very exciting to see that the seeded fibril with this mouse PHF have distinct pattern. Now, in this publication, we say we are really not ready and talk about what those structure correspond to, but clearly the seeded fibers have narrow and distinct distribution, but not a single one. So this is something a lot more studies need to be done. Now, in an ongoing study, I can also report that in vitro seeding with cellular material that have been inoculated with human brain material in collaboration with Amanda Wehrman. Initially, it was a collaboration also with Proteinopathy, but Amanda is at UMass, a professor there. It is the same story. It really works. You take the cellular material, you can achieve seeding. But the second generation is weaker unless what I called magic is needed. So we definitely need additional cofactor, same idea, at the amount that in and of itself cannot induce aggregation. And interestingly, we also needed DTT. Now the DTT isn't on the tau incubating material, so it's nothing to do with disulfide bond of the tau itself, but there's something about the seed that requires some of this. I say magic just because a black box, and it has been reported that DTT is needed, but it's not clear why. So same idea, we have activity, but then achieving structural conversion is a different story, and here we are also making progress. So far, what I have shown you is that there is perhaps something like a potent cofactor, for example, heparin, that can very rapidly and very efficiently induce aggregation, but does so in a non-templating way. It's fast, it's spontaneous, there's no annealing, you have a heterogeneous structure, unless certain procedures are used. Then you could have a mild cofactor, for example, RNA, that just lowers the barrier, but then the seed needs to be the one, seems to be the one that then completes this cycle, and perhaps, at least so far, our results show, achieve some amount of structural convergence. Now, it is important to overcome the seeding barrier in various ways. So with that, I think I can slowly transition into the role of LLPS. So before we go there, though, we were asking the question, so what does this so-called mild cofactor RNA exactly do to tau? What we have found is that RNA and tau, when we incubate them under condition that induce seeded aggregation, it's not just a molecular complex that form. It does seem to form oligomers. So you see here the DLS studies with protein and poly-U. It's certainly more than a molecular size. We can see by electron paramagnetic resonance that protein in the presence of poly-U is significantly mobilized. And when we do cryo-TM of the soup that has the RNA and tau complex, then we certainly see consistently species that are more than a molecular complex. So RNA and tau form some version of stable oligomer complex of origin that is not entirely clear. So this tau RNA complex that we see is, however, stable. Stable in the sense that it doesn't convert to anything. So you saw there's this species that formed that we can clearly see by DLS, EPR, and TM, but they stay like that until THG active fibril species in the form of a seed is added. So I'm showing you here that only when you have those complexes, then you add a very small amount of seed that in and of itself can't do anything. But in that context, THG activity shoots up. And then when we chew up the RNA with RNase, you see then that activity is gone, okay? So it looks like tau RNA complexes that are beyond just molecular complexes can be effectively converted to fibrillar species. That is a good segue from me to talk about liquid-liquid phase separation. So we knew from the collaboration with Ken Kosick and May that RNA naturally associate with tau in the cell. And we discovered concurrently that RNA and tau interaction readily promotes liquid-liquid phase separation. So this is a study that some of you might know. I'm showing you just a movie to make it clear that these are really droplets, and we have droplets merging and dynamically moving. And we have shown earlier something that wouldn't be a surprise to those who study LLPS, but their tau is really exclusively inside a droplet. Just a few words about tau. If you remember, I said there's an N-terminus that is slightly negatively charged. The polyproline rich domain is positively charged. The microtubule binding region is also positively charged. And the C-terminus is mixed, but slightly still a certain part is positively charged, the very end is negatively charged, okay? So you can say tau overall is a polycation. And so it associates with polyanion, and the LLPS is particularly effective at a net one-to-one charge ratio. Now, when we initially did these studies, the salt concentration as well as the tau concentration were not necessarily physiological. So in a follow-up study, we were able to show that, first of all, this droplet formation is enhanced at high temperature. It shows what is known as lower critical transition temperature, LCST behavior, solution temperature behavior, LCST behavior, and that is entirely reversible. Okay, some of these changes here is due to degradation of RNA, but the process itself is entirely reversible. We were also able to show, because here it's an entropy-driven process, we can now achieve much lower concentration of tau to induce LLPS for example, by going to 37 degrees Celsius as opposed to 25, and by using crowding agent, depletant agent, for example, PEG or dextran. And so in that study, we entitled our study, the title was Narrow Equilibrium Window for Complex Co-Conservation of Tau and RNA under Cellular Condition. And the idea was that both computational studies done by Joan Shea and Glenn Frederickson group and the experimental group that we did showed that we really are under physiological condition of salt concentration, temperature, as well as tau, right at the cusp where readily LLPS condition can occur. And on the left-hand side, this is not even taking crowding into account. And if now additionally you have crowding forces that can be exerted by macromolecules or salt, then that concentration at which LLPS can occur might be even further dropped than a few micromolar. And so obviously the question then arises, what about in live cells? I'll get there. But first, before I do that, of course, the burning question then is, what is the relationship between this liquid-liquid phase separation and aggregation? So when we were focusing on complex co-conservation between tau and RNA, okay, what we found is under those conditions, and I have a qualifier in there because I'll show you in a couple slides. Under other conditions, the result is different. But anyway, if you do LLPS between tau and RNA, and we look at carefully the THD fluorescence, which is a proxy for fibril formation, and turbidity, which is a proxy for droplet formation, that the conditions overlap, but the two processes are distinct processes. Okay, we have done a lot of studies to show there are distinct processes. I will highlight just a couple. So for example, when I look at the aggregation half-time as a function of sodium chloride concentration, in which the red is showing droplet condition and the black is non-droplet condition, you see that there is no sudden change, but there is a contiguous change. Or when I look at the aggregation half-time as a function of heparin concentration, and we know that there is a certain optimum ratio at which aggregation at around 4:1 ratio is particularly aggressive. Again, looking at it at two different salt concentration, one droplet condition, one non-droplet condition, there's just a continuous change. It is clear, looking at it carefully, that the liquid-liquid phase separation of tau in and of itself doesn't promote or facilitate aggregation. So not just the act of droplet formation itself isn't sufficient. And we looked at the monomer dependence of the half-time too, but perhaps I will skip that in the interest of time, but perhaps highlight just one more study when we look at the wild type versus P3ONL. And I mentioned earlier that the P3ONL mutation is one of the most widely used and famous disease mutation because it's so potent in inducing aggregation. I can report that the liquid-liquid phase separation propensity is indistinguishable, but of course, as we knew, the fibril propensity is very different. Now, that was, so we concluded that electrostatically driven complex co-conservation and amyloid aggregation of tau are independent processes with overlapping condition. But there are other ways to generate LLPS. We can liquid-liquid phase separate tau without RNA, as has been also reported. Actually, in fact, recently, Markus Zweckstetter gave a nice talk in the ZOOMinar, and we know of work of several other groups that do important studies in this realm. But I can say that taking tau, especially full-length tau without RNA, we can induce phase separation without RNA, either by going to very low salt concentration, and it does something called simple co-conservation, where the charge of the tau itself does the job. Or, and this is the important part, we can go to very high salt concentration, extremely high, 3 to 5 molar. And you see here that we can induce liquid-liquid phase separation. So it will become clear why I'm even mentioning this study. What was really stunning to us is that the complex co-oscillation that was induced by electrostatic co-oscillation between tau and RNA were insensitive to 1,6-hexanediol. And I'm mentioning this 1,6-hexanediol molecule because for in cell as well as cellular condition LLPS in a biological context, it has been used by many to show that your biological condensate is a liquid and that you can basically melt it. But the complex co-activated tau with 1,6-hexanediol didn't care. You couldn't melt it with this hydrophobic molecule. But the high salt droplets are entirely different. You can indeed melt the high salt droplet by adding a certain percent of 1,6-hexanediol. It's a hydrophobic molecule. And as you see on the right-hand side with the THD curve, at the moment you abolish the liquid-liquid phase separation, also the fibril propensity is entirely abolished. So different types of droplets are formed. High salt, long story short, induced depletion interaction induced droplet with where the tau molecules are held together by hydrophobic interaction. And that's why 1,6-hexanediol can be effective. So now to the relationship to fibril. We can show here visually as you induce more and more droplets, indeed that correlates exactly with fibril forming propensity. I'm showing here also TM of the fibril, but perhaps this graph is most efficient. It's plotting the initial turbidity at incubation time 0, meaning the amount of droplet formed, versus the final THD indicating the final fibril amount formed. And you see a very strong correlation. And I contrast that to the tau RNA complex co-activation shown here in a square that had no such dependency. There is a clear dependence. So LLPS driven by hydrophobic interaction facilitate transformation of soluble tau to fibril. A side note, but as a very interesting side note, we see that the P3NL mutation now of the same 2 and 4R full-length tau has much greater propensity to form fibril from, as well as much greater propensity to form droplets. That's because the hydrophobic interaction is much greater between P3NL tau compared to wild-type tau. So why that is, that's a different story. And we have done a lot of studies to verify that indeed the hydrophobicity is different. And finally, if some of you will think high salt at 3 to 5 molar, how is that biological? That salt effect isn't biological, of course, but the effect that it can exert can well be biological. So for example, the high salt basically entirely screens the charges, and that might be a mimic for what post-translational modification could do by acetylation or phosphorylation that mute a strong positive charge density from the abundant lysines around tau. And of course, the crowding pressure effect can be mimicked by high salt condition, especially because this effect follows the Hofmeister series. And so finally, I have to come to this point. So how about in the cell? So I would like to feature in the last couple slides work done by Ken Kosick and Mei Zhang. So they utilized the concept pioneered by Brangwine with the so-called optodroplets by adding the optically active Cry2 that upon blue activation can induce association together with 2 and 4R tau. And they showed in this very recently published paper that the microtubule binding of tau upon blue light activation can be enhanced. But of course, what they wanted to see is the droplet. So here you can see the higher density here, but you can't see exactly droplet. So what's really interesting is as long as you have the microtubule binding repeats there, either as part of just a binding domain, MTB domain, or the full-length tau, you see effects but not droplets. And that entirely changes when you just look at the polyproline-rich domain. Now you see droplet when you have up to PRD tau. And upon blue activation and you turn it off, you see that the droplets disappear, or when you have it on for a long time, they evolve. So LLPS of up to PRD tau is reversible. And here all shown together, 1 to 441, so full length, then just a microtubule binding repeat, and just a PRD region, PRD plus the microtubule binding repeat. You see that you have activity either when you have the full length tau, you see enhancement upon blue activation right here, or you see in particular enhancement when you have the PRD domain driving. And so in-cell droplet formation is postulated to be driven by polyproline-rich domain because they are not occupied by the microtubule binding activities. And a side note, interestingly, the hyperphosphorylation of the specific PRD domain appears to abolish LLPS, and that would be consistent with perhaps a negatively charged cofactor is involved in the LLPS of PRD, because then hyperphosphorylation would diminish that interaction with RNA. And this is just to say, in vitro, we verified, and it's the biology bit, meaning really the LLPS itself between the MTB region and a PRD is not that different, but really it's the biology, how certain repeats, for example, interact with a microtubule that made all the difference. So in conclusion, I have given you a long-winded way of saying we still don't know how to converge the dynamic conformational follow the achieve structural convergence, but hopefully I have shown you that we made progress with seeding assay, how to induce effective seeding in vitro, how liquid-liquid phase separation can be an active container under certain condition, but pathway selection is still something that we and many others are working on, and it's an important goal. With that, I need to finish with an acknowledgement. My postdoc, or former postdoc, Dr. Jan Fischu, did a lot of this work, Michael Weigers, Yan Chan Lin, Kate Zhang, and also Shiny Mighty. And I think I mentioned all my collaborators. So with that, thank you very much for your attention.
Thank you very much, Songi. So we're going to move to the question period, and I'll ask the participants to put your questions in the Q&A box, and then we will admit you to the main session, and you can ask your question to Songi directly. So because
should I stop sharing?
No, you can keep it in case people have questions, but it's up to you.
Yeah, okay.
Bikash, could you let Galina and Christian into the room? And I see Ben is already there. Ben, would you like to ask a question?
Yes, I do. Can you hear me?
Yes.
Yeah, great talk, Dr. Professor Han. I have a question related to the cofactor in the tau aggregation. So I think you mentioned RNA. So does the RNA do you have to specify, the length or, the sequence? Is there any dependence on that? And can you comment whether it's really the charge? Because the one common feature for RNA and heparin is they both have a negative charge. And there are also some reports saying bacterial DNA can promote aggregation. So I was wondering, all these cofactors, the RNA, DNA, or maybe some others, maybe potential phosphorylation, all this relate to, the charge essentially. Can you comment on that?
Yes. When it comes to liquid-liquid phase separation, certainly it's the charge. And we know that because RNA, DNA, especially single-stranded DNA, and many other polyanions can induce LLPS pretty indiscriminatorily. We also know from other work and our own that hyperphosphorylation that changes the charge will shift the optimum liquid-liquid phase separation regime. Now, but specificity plays a role when it comes to then aggregation. When it comes to aggregation, it's not just a charge. And so there, it's harder for me to answer because when it comes to the seeding assay, we have so far had good success with RNA. I can say so far the sequence, we tried a little bit to look at the sequence specificity, didn't find something obvious, but doesn't mean it's not there. So I think LLPS is a much more general phenomenon, a robust phenomenon. When it comes to aggregation, it's a much more specific event that needs delicate balancing of these factors.
I have a second question about your technique. I think this is a great technique about the DEER, your EPR labeling. Can you compare this to like a FRET, for example, because they are both like measuring the distance, whether the FRET can do the similar thing, or what's the advantage of your DEER method?
Right. So the advantage of FRET is certainly it's much more sensitive, which is why the biosensor cell rely on FRET. But FRET without single molecule technique will only give you something like a mean distance. Now, single molecule FRET can give you a distribution, but the statistics isn't large enough and the distance isn't accurate enough to really give you a shape. It will give you a distribution, but not in the same way DEER can do. So DEER is less sensitive, but it gives you much greater detail of structure as well as the complete ensemble, I would argue.
Thank you.
Thank you, Ben. So Galina, you can unmute yourself.
Hi, Dr. Han. Excellent presentation, and thank you very much for putting everything in very succinct terms that we need to look for the convergence of the aggregation basically within tau. That's something that we managed to develop in our lab as well in vitro. So I would like to ask more of a technical question about the DEER. So with respect to labeling and timings of incubation and also the cofactor use, so what sort of cofactors you use with a heparin molecular weight, for example, and how long does it take to label it, and sort of from this part, and also what are the dimensions of the fibrils that you get from the RNA induced or heparin induced fibrils, please?
So the labeling itself requires, of course, recombinant expression of tau with deliberately inserted cysteine. That itself isn't difficult, but we need to be very careful of where we put the cysteine because, for example, we don't want it to interfere with the fiber. But yeah, recombinant expression and labeling with well-established protocol is not particularly difficult, especially because tau expresses generally quite well. In terms of seeding, we so far used just RNA, and because we didn't immediately find any RNA sequence dependence, we just used poly-U because it's a convenient cofactor. And the criteria is that we use a mild cofactor. We don't like to use heparin because heparin is so effective itself. The cofactor is in the way of the seed. So we like using cofactor that associate but doesn't seem to induce structural convergence. What was the other question again? Did I answer everything?
The dimensions of the fibrils?
Oh, the dimension of the fibrils. So I would like to say that seems to depend more on the tau that we use than on the cofactor. We don't do the fibril analysis as sophisticated as we should. We just use TEM analysis, and it's, let's see, I mean, maybe micron long fibril, but the quality of the fibril actually depends a lot on the exact tau type used, almost more than the cofactor. But we haven't investigated that.
Are these single fibrils or are they doublets?
That depends also on the structure. We are trying to figure out a way to actually determine whether it's single or double, also by spectroscopy. But yeah, I cannot answer that other than we sometimes see single, we sometimes see double.
Excellent. Thank you very much.
Mm-hmm.
Thank you, Galina. And Christian, can you unmute?
Yes. So Christian Grisinger here. Hi, Songi.
Hi. Hi, Christian.
Very nice talk. Maybe it escaped my attention, but you mentioned this, that with the RNA, you could have more than one round of seeding. Mm-hmm. Now my question is, were you then able to reproduce one of these post-mortem brain seeds looking at the distances with Peldor, or is that still kind of an outstanding goal?
It's outstanding. So I can tell you the following. We are trying very hard to, even in the first round, to get the daughter fibril motif measured and see whether it replicates the mother fibril motif. And I can tell you already that we see certain distances that are there in the daughter fibril, but we see multiple distances. So in other words, even in the first generation, we haven't achieved convergence. Now, when we do a second generation, maybe we can clean up the fibril and converge, but those are ongoing studies. This is, I think, on everyone's mind. I'm sure on yours too, but we haven't achieved it yet. No.
I was wondering. I mean, Chris Zironiak, I think in a talk I remember on prion, observed similar things, that if he takes a certain seed, then it does not reproduce necessarily that seed. But are you sure that the seed actually has a unique distance distribution, or is that also unknown?
It is unknown. Here's what I can tell you. We are getting seeds from our collaborators that are originally human tau derived, but then they go through rounds of inoculation via the cell. By the time it passes through the cell, the material itself has evidence of activity. However, nobody knows the structure. So I'm hoping that from the cryo-EM experts who might be listening or not, that structures of the seed from the cell is desperately needed, but structures of this are not known yet.
Okay, thank you very much.
Yeah.
Thank you, Christian. So Songi, if you go to the Q&A box, you'll see the fourth question is by an anonymous attendee. So if you would like to read
I'll read it. Okay.
Out loud.
So you have evidence for the important role of anions in facilitating tau aggregation. In the case of heparin, do you think the relative spatial separation of anions play a role in organizing lysine residues in tau and templating amyloid formation? So let me answer that in the way I can. There's a really good question. One thing I can say is that one big difference between heparin and RNA is the persistence length or the stiffness of the polymer. So I absolutely do believe that the heparin backbone stiffness plays a role in templation. Second, we know from our work that others, I believe, also published, but we also have investigated, namely heparin versus 6-O desulfation, N-O desulfation, 2-O desulfation. We should be changing the desulfation degree of heparin, which inevitably would change the spacing, have very big effect on aggregation. And so judging by which sulfates on the heparin is sulfated seems to play a very big role in aggregation. Kinetic suggests to me the answer is yes. Both the spacing as well as the stiffness of the template appears to matter. That's the best answer I can give.
Thanks, Songi. So our next question is from Vijay.
Hi, Songi. This beautiful talk. Can you hear me?
Yes.
Okay. I have two questions. The first one is regarding the probability distributions that you've noticed, and I'm wondering whether you were able to do this in a temporal scale as a function of time. And if so, do you see a change with time towards a specific bias of a particular type of an ensemble? And have you seen these probability distributions in liquid-liquid phase separation conditions as compared to the aggregation conditions?
Yeah, really good question. So for the first one, I can say, when earlier in one of the slides, I talked about how we tracked the PHF6 stretching. So there we indeed took the temporal scale. We freeze quenched at 1 minute, at 10 minutes, at 1 hour, and at 12 hours. And we were able to see that, for example, this conformational extension that seems to make the tau aggregation prone happens very early. Now, the same technique to look at, for example, seeded aggregation, we haven't done yet. But the reason we haven't done that yet is that the seeded aggregation is much harder than just dumping heparin on the tau. And so we first need to make sure that when we have several hours of aggregation, we have a distinct pattern. And then, of course, we then need to figure out what look, we are dying to know what part folds first and what parts folds later. That's the dream. But to get there, we need to have, first of all, 1 or 2 structure. Right now, we have a mess of a structure. And I would argue everyone has a mess of a structure, if I may say that. So that's the goal. The second question about the cofactor, do you think the cofactor? Can you repeat the second question?
Well, I wanted to know the cofactor requirement for seeding. Is this a tau-specific phenomenon? Because A beta does not require such cofactors.
Yes, I would actually think so, because as many people actually in this, even in this round or in this ZOOMinar has mentioned, tau is incredibly stable. Tau is just rock solid. It doesn't aggregate, actually. So you have to do something to tau for it to aggregate, because it just wants to be happily dissolved as an IDP. So I do think it is somewhat tau specific. I already know from alpha-synuclein that you can incubate it without cofactor and could converge to structure. I think the other question you actually had was the PRVA in LLPS. So I can say that we learned that it depends on the type of droplet. So if you have this fully reversible droplet that seems to just make droplets and then just be very not doing anything, then the PRVA doesn't change. At the moment, we have the droplet that seem to age and aggregate or induce aggregation. We indeed see conformational changes.
Right. Thank you.
Thank you, Vijay. So we're up at the top of the hour. So I'd like to first thank Songi for an absolutely fantastic talk. And also I'd like to invite those who'd like to talk more one-on-one with Songi to join our open discussion now. So the formal session is over, and Bikash is going to admit anyone to the main session who would like to chat a little more with Songi. And Songi, maybe you can stop sharing your screen now. Thank you so much.
Songi, I have a question to ask.
Hi, Rams.
Great talk, great talk. I have a technical question. So how about the mobility of the protein itself? Does it have any influence on the distribution that you measure, in addition to having an influence on the measured dipole-dipole coupling?
So the way I would like to answer that question is that
the time scale of mobility is entirely different. Doesn't influence the EPR measurement? I don't know. Just
So from that point of view, if you mean literally dynamic as in movement, the DEER measurement are vitrified. So the EPR line shape measurement we can do in a dynamical state. So we can see the ends are mobile, the middle is stiff. Those things we can do in room temperature as well as a function of temperature. So CW EPR line shape analysis can be done under ambient condition. But DEER, with the dipolar coupling, it's like in NMR, in order to have dipolar coupling, you need to not have the distance average out. So it's vitrified, a capillary is vitrified under liquid nitrogen or even helium temperature.
Oh, it's okay. Sorry. Yeah. What about the distribution of distances due to mobility?
Yeah, same idea. So
it's frozen. Okay. Yeah.
It's frozen, but of course, the convergence is important, and so we are, I think you know what I mean.
Very nice talk. I have a question. Like in one of your slide, I think you have shown the short fragment of tau, and when you have a mutation from proline to leucine, you could see a very fast aggregation kinetics based on fluorescence, and the amount of fibril also you got a lot, right? So I was wondering, like the proline to leucine mutation, is it like biologically relevant, like any kind of familial?
Oh yeah, very much. So are we off record? Then I can tell you. Well, doesn't matter. Okay, off record, on record, doesn't matter. So it's very biologically relevant. Every single mouse model, including the most recent one from 2020, published by Virginia Lee, relies on what is known as the P3-on-S or P3-on-S-L mouse. Every single cell model by Mark Diamond, by anybody, has the P3-on-L or P3-on-S mutation. There is no model in tau that doesn't use the P3-on-L or S mutation. Okay, so it's a very, I would say, the most important mutation besides a couple other that people use, such as V337M. So what's really interesting is that you become smaller and smaller and smaller, and you get down to this 20 amino acid fragment. It's still the P3-on-L makes a difference. So we do think there is both conformational bias and hydrophobicity involved. And P of R showed us that the initial P of R is actually very similar, but at the moment you initiate aggregation, the hydrophobicity and the behavior of the protein is very different. So we are still trying to figure out what this is. We do think the hydrophobicity is different. Why?
Yeah, I think that is pretty understandable at a point of like very short fragment, because proline is known to be a kink breaker. Like if you have some kind of alpha helix or something, it can like, the intermediate structure is somehow affected when you convert proline to leucine. But I was wondering in a like long time point, like if you are taking the full length of tau and you have this mutation, I'm wondering how this single mutation can actually affect.
It seems to. I can tell you empirically by us and others, whether you take the full length tau, 441 residue, half the tau, middle of the tau, small tau, doesn't matter. This one mutation has dramatic effect. So all the way long, all the way to small. And so, but interestingly, it seems to have an effect at the moment you try to pack it. It's not so apparent in the IDP state.
Right, thank you. I think Anup has some question, I can allow him.
Songi, you in your THT measurement, does it measure only the beta sheet content, or does it measure the droplet formation as well? I was a little bit confused in one of the plots.
So we take THT as a proxy for fibril, and we take the turbidity together with microscopy as a proxy for droplet.
THT also, does it give any turbidity measurement or nothing?
No.
Okay.
Anup, you can ask your question.
Hello.
Hello.
Hi. I was very curious about this DTT data that you showed. I was thinking, like in general, when we use the reducing agent, it will cause some structural changes, right? Because it's going to interact with your disulfide and all that. But in this case, you propose that as if it's seeding. It can be like used as a seeding at this concentration that you mentioned. So,
I should clarify. I should clarify what I meant. I meant that when you induce aggregation with a seed material derived from cell or say a mouse model, that we knew from the protocol from others that they mentioned DTT. So we were just curious and tried and found that you need to apply DTT to the seeding material in order for the seeding material to be effective. We don't know why. And I can tell you that the DTT effect isn't on the tau that we are trying to aggregate. So something about the seeding material perhaps needs a reducing agent to become activated. Now I'm speaking like a black box, so we don't know why. But it's just one of those many things when we work with biological material, that's what I meant with there is some amount of magic involved that we don't understand, such as we need to apply DTT to the seeding material.
I see. Okay, based on what you just mentioned, it looks like it's something to do with the free thiols available in both species, which is kind of making it possible, the magic.
Yeah.
All right. So yeah, but overall, it's a good talk. Thanks.
Mm-hmm.
Hey, Songi, this is Vijay again.
Hi.
Hi. I just, one of the things you mentioned in one of the slides is that the LLPS, the hydrophobic interactions driven LLPS, if I'm right. Mm-hmm. And I'm wondering, I mean, isn't this goes against the phase separation phenomenon where you have a very high dominance of hydrophobic interaction eventually will shear away from phase separation? Isn't it counterintuitive thing?
Yeah, so I would say, I start by saying I've worked on LLPS initially not because of tau, but because of other proteins and other polymers. And I would argue you can make droplets out of almost any protein, I would say, as long as you can somehow make it to weakly and multivalently interact. So the hydrophobic interaction in this case would be, so you have tau, they have a lot of charges, and by adding a humongous amount of salt, first of all, you screen the charges. But then there appears to be weak, potentially multivalent van der Waals interaction together with the water basically getting excluded from the interior that induces LLPS. So I would say you can induce LLPS by any attractive interaction. It could be charge, pi cation, hydrogen bonding, right, but also hydrophobic interaction. Yeah. So But
if you tend to have more bias towards tryptophans, you normally do not see phase separation. Rather, you see the more aggregation type phenomenon, isn't it?
I see. Yeah. That's if the hydrophobic interactions are too strong. You're right. I mean, there's a balance when when other interactions take over. They dominate over the LLPS because you need weak long range interaction. So yeah, you're right. But tau doesn't have that many. I mean, right, tau is relatively not so hydrophobic. Doesn't have arginine and many dose of residues. So I think for a weakly hydrophobic interaction, it works.
Got it. Thanks.
Vikash, we have a lot of people to admit.
Yeah, I'm doing one by one.
Oh, sorry. Okay, great.
Hi, Songi. I have one more question about the various parts of the tau, their contribution to aggregation. So you study, of course, mostly on the repeat structure, R1 to R4. Have you done anything, related to the N-terminal, N1, N2, and the protein-rich region, whether they contribute in any way to the tau aggregation? And related to that, you mentioned the R1 through R4, that is the cron EM structure actually covers extended from R4. That is a region which is not, just R4 that I think some people may call R5 or something like that. Does that make any difference?
Yeah. So first of all, the N-terminus is considered by, and there are studies by others, also, of course, explicitly to be rather unhelpful to aggregation. I don't know of any studies where the N-terminus is part of the structure and cutting off any part of the N-terminus helps, but we haven't specifically looked at the role of how much of the N-terminus plays a role. Now, the polyproline rich domain would be right outside the core. I do secretly think it's very important, but I cannot prove it. So we do want to work on the role of the PRD domain in aggregation. It could be helpful, but certainly PRD won't stack into beta sheet. Now about the extension beyond R4, certainly. So this was one of the surprises when the cryo-EM structures came out, that the core extended to something like 394, which is beyond the traditional R4 designation, right? Mm-hmm. We do think it's very important. And you can kind of see it when you look at the cryo-EM structure that is butting against this other, you know, it's stabilizing part of the core. But again, I think, yeah, there are so many degrees of freedom to study, but yes, we do think it's very important.
Thank you.
Bina?
Hi, I have sort of a follow-up question from the last person. Has there been a deletion analysis wherein probably you get rid of the N-terminal, the C-terminal, you take only the repeats, and then see how the cofactor affects? Because eventually the cofactor is not part of the core of the aggregate. So is it that it initially binds to certain other regions of the tau and then sort of initiates this aggregation process?
Mm-hmm. So I'll try to only answer the part I know. So the N-terminus, I really don't know whether having it and not having it then affects the role of cofactor. I can imagine, just because the N-terminus is negatively charged and maybe it plays a role, but at least on the surface, the N-terminus seems to be rather just in the way. About the cofactor, indeed, on the one hand, the cryo-EM structures don't have the cofactor well, but remember the CT and CBD structure have the cofactor in it. Even though it's not clear what is the cofactor, at least in a CBD case, the Sherris and Goddard group postulated it might be polyanionic. So it's possible. I can tell you only that from our in vitro studies, when we work with cofactor and then you take the fibril, you clean it and you look at it, and then you apply RNase or heparinase to melt the cofactor away, it plays a role. So at least in the in vitro studies, unless you do something different, the cofactor actually stays associated. That's what we think.
Okay, thank you.
Thank you, Bina. Sahil?
Hello.
Hi.
Hi. So you said that you see when the fibrils are formed with the RNA and with heparin, they are different. So when formed with RNA, they have a less persistent length.
Oh, sorry, there was a misunderstanding. I meant the persistence length of the RNA and heparin itself is different. RNA is floppier and heparin is stiffer. And okay, so I was saying the heparin is more effective because of that.
So the fibrils formed have same morphology or have different?
Yeah, we are not experts in fibril morphology. I really cannot answer that. I think they are really, yeah, there are probably other people who can answer that better. We don't know. And I was told by at least the Goddard group, don't trust TM too much and don't read too many tea leaves. So we don't know. We really can't answer that question.
Thank you. Galina?
Hi. So is the mechanism of RNA-induced tau aggregation known? For example, which residues are the binding ones and whether it is predominantly due to charge neutralization, but also maybe direct conformational change of tau structure on local or global scale?
So I would say it's not known, but I can tell you just what we think is going on. At least our hypothesis is that tau and RNA associate with each other initially non-specifically. Why we say that is because when we look at the complexes, they appear to be just dynamic blobs. So we do think that the tau RNA association, at least initially, is non-specific charge-charge interaction. It requires a seed in the form of a mini fibril for then that sort of non-specific complex to turn into fibril. So my hypothesis would be that RNA lowers the free energy or some sort of barrier, and that when the seed comes in, that is the actual agent that transforms fibril, that then templation happens by the seed. That would be my hypothesis, but I don't know, and I don't think that, at least no published study about that specifically, as to the best of my knowledge.
Yes, exactly. There's not much in the literature so far. Thank you.
Yeah.
All right. Well, it's past 8:15 now, and Songi, I want to thank you for staying longer to chat a little more, and certainly thank you for the talk. It was brilliant, and I think we'll wrap up the session now. So your recording will be posted shortly. I guess we will let everyone know when it's posted.
Okay. Thank you.
Thank you very much, Songi. It was a great talk. Thank you very much.
Thank you. Thank you very much. Yeah.